Let's start with Streptococcus, a fascinating genus of bacteria that are spherical and tend to form chains. These bacteria have a distinctive appearance that really sets them apart. Understanding their structure is key because it influences how they interact with their environment and hosts. This introduction sets the stage for exploring their characteristics, classification, and clinical importance. Now, what defines Streptococcus? They're gram-positive, meaning their cell walls have a thick peptidoglycan layer that retains the stain. They don't form spores or move on their own, which affects their survival strategies. Interestingly, they can form capsules that protect them from the immune system. They're facultative anaerobes, so they can grow with or without oxygen, but they are fastidious, needing specific nutrients. Their colonies are small and sensitive to environmental factors, which helps us identify them in the lab. Under the microscope, Streptococcus cells appear as purple-stained spherical chains, confirming their gram-positive nature. On blood agar plates, their hemolytic activity is a key diagnostic feature. Alpha hemolysis causes partial breakdown, creating a greenish zone. Beta hemolysis leads to complete clearing around colonies. And gamma hemolysis shows no change. These patterns help differentiate species and guide clinical diagnosis. Classification of Streptococcus hinges on oxygen tolerance and hemolytic patterns. Aerobic species grow on blood agar and are divided by hemolysis. Alpha types like viridans and pneumonia show partial hemolysis. Beta types, grouped by Lancefield antigens, exhibit complete hemolysis. And gamma types, such as Enterococcus faecalis, show no hemolysis. Anaerobic species include Streptococci. This framework is crucial for identifying and treating infections. Focusing on human pathogens, several Streptococcus species stand out. Group A Streptococcus, or S. pyogenes, is a major cause of infections. The viridans group includes species like S. metis and S. mutans, important in oral health. S. pneumonia is known for respiratory infections, and S. faecalis, or Enterococcus, is notable in nosocomial infections. Recognizing these helps us understand their clinical impact. Let's dive into the virulence factors of Streptococcus pyogenes, which explain its ability to cause disease. The M protein is key, preventing phagocytosis and aiding attachment to host cells. Lipoteichoic acid and F protein assist in binding. Streptokinase helps the bacteria spread by breaking down clots. DNAases reduce pus viscosity, aiding movement. Pyrogenic exotoxins trigger intense immune responses. The capsule shields from immune attack, while hyaluronidase breaks down tissue barriers. Hemolysins destroy blood cells, and peptidoglycan contributes to inflammation. These factors work together, making S. pyogenes a formidable pathogen. Turning to Streptococcus pneumonia, its virulence relies on four main factors. The capsular polysaccharide prevents phagocytosis and is the target of vaccines. Pneumolysin disrupts mucociliary clearance and immune cells. Autolysin causes bacterial cell wall breakdown, releasing inflammatory components. IgA1 protease cleaves antibodies, helping the bacteria colonize mucosal surfaces. Together, these factors enable the bacteria to evade defenses and cause serious infections. Understanding Streptococcus pathogenesis involves several steps. Humans are the exclusive reservoirs, meaning transmission occurs only between people. The bacteria spread through contact and respiratory droplets. They typically enter via the skin or throat, with children being the most affected group. If untreated, infections can lead to serious complications, emphasizing the importance of early detection and management. Group A Streptococcus causes a range of diseases. Pyogenic infections include respiratory illnesses like tonsillitis and scarlet fever, skin infections such as impetigo, and deep infections like septicemia. Non-suppurative conditions include acute glomerulonephritis affecting the kidneys and rheumatic fever, which involves inflammatory heart damage. This diversity in clinical presentation highlights the need for careful diagnosis and treatment. Rheumatic heart disease develops through a complex immune process after Streptococcus infection. Antibodies produced against M proteins can mistakenly attack heart tissue, causing inflammation and damage. This cross-reactivity leads to chronic valvular heart disease and increases susceptibility to bacterial endocarditis. Understanding this mechanism is vital for preventing long-term cardiac complications after streptococcal infections. Post-streptococcal glomerulonephritis is a kidney disorder that develops after a strep throat or skin infection caused by Streptococcus bacteria. It's characterized by inflammation of the glomeruli, the kidney's filtering units, leading to symptoms like blood and protein in the urine, swelling, and high blood pressure. PSGN is a type of post-infectious glomerulonephritis and is most common in children. Treatment typically involves managing symptoms like high blood pressure and fluid retention with medications and treating any underlying streptococcal infection with antibiotics. Risk factors for Streptococcus pneumonia infections mainly include age, the immune status, and presence of other complications and infections. The bacteria usually multiplies in the oropharynx, initially giving rise to inflammatory response by the host. It can also lead to middle ear infections after gaining entry into the middle ear through eustachian tube. Other diseases include lobar pneumonia, bronchopneumonia, and even meningitis. Streptococcus mutans is a bacterium strongly implicated in the development of dental caries, commonly known as tooth decay. It's a primary contributor to the formation of dental plaque, a biofilm on tooth surfaces where it produces acid from sugars, leading to enamel demineralization and ultimately cavities. Laboratory diagnosis starts from sample collection. In this case, it can be PUS, swabs, or blood, depending on the site and type of infection. First test that follows is staining methods, where they appear as gram-positive cocci in chains in gram staining. However, confirmation of organism is achieved through culture, where they require capnophilic condition. Species identification is achieved through tests like bile solubility test, negative staining, and bile escalon agar test. The CAMP test is a microbiological test primarily used to identify Streptococcus agalactiae, group B Streptococcus. It detects the presence of a protein called the CAMP factor, which, when combined with the beta-hemolysin produced by Staphylococcus aureus, enhances the lysis of red blood cells. This enhanced lysis is visualized as a characteristic arrowhead-shaped zone of hemolysis on blood agar. Serological methods are vital for Streptococcus identification. Lansfield grouping classifies bacteria into groups based on cell wall carbohydrates. Griffith typing further categorizes group A strains. Capsule swelling reactions identify encapsulated types. Agglutination tests detect antibodies like ASO and CRP. Enzyme immunoassays and immunofluorescence techniques provide sensitive antigen detection. Together, these methods offer a comprehensive approach to diagnosing streptococcal infections. When it comes to treatment, beta-lactam antibiotics like penicillins and cephalosporins are frontline options due to their effectiveness. Quinolones inhibit bacterial DNA replication, offering another mechanism. Sulfonamides combined with trimethoprim are generally ineffective against Enterococcus due to intrinsic resistance. Glycopeptides like vancomycin are crucial for resistant strains. Understanding these options and their limitations guides effective clinical therapy.